So under denaturation, so what do you mean by denaturation? So this is a double-stranded DNA. So this red color, it shows the region that is to be amplified, or whatever the sequences that we want to have it in more copies, right? So this is the double-stranded DNA. So the first step is the denaturation, that is heating of this target DNA at high temperatures, that is about 94 degrees centigrade, to separate the strands. So now the double-stranded DNA is separated to form the two single strands. Now each of these strands, it acts as a template for DNA synthesis. So after denaturation, the double-stranded DNA is converted to single-stranded DNA, and now these two single strands, they act as a template. Then the second process is annealing. So in annealing, it is the joining of two primers. So this is a primer, and this is another primer at the 3' end. So this is the 3' end, and here for this strand, this is the 3' end. So the joining of two primers at the 3' end of the DNA template, and this process is called annealing. So here the temperature is less, that is about 40 to 60 degrees centigrade. So there denaturation at high temperature so that the two strands get separated. Here the joining of two primers at the 3' end, that process is called annealing. Then the third step is the extension. So what is the extension? It is the addition of nucleotides to the primer. So already there exists a primer. So now there occurs an addition of nucleotides to the primer using a thermostable DNA polymerase. So this is taking place at the higher temperature, that is about 72 degrees centigrade extension. So by the help of the enzyme that is called tag polymerase. So this tag polymerase, actually it is different from the other enzymes in that it is resistant to higher temperatures, and it is obtained from the bacterium that is called Thermus aquaticus. So that is named as tag polymerase. So it remains active in higher temperature during the denaturation of double-stranded DNA. So even during the denaturation, this enzyme is active even at a higher temperature. So through continuous replication, the DNA segment is amplified up to 1 billion copies. So even up to 1 billion copies, the DNA segment can be amplified, or it can be produced in more numbers. So this amplified fragment can be used to ligate with the vector for further cloning. So you can get that particular fragment of DNA in more copies. So I think you can understand this diagram, right? So this is a double-stranded DNA. On denaturation, it forms two single strands, and then joining of two primers at the 3' end. Then there occurs an extension, that is the addition of nucleotides. Again, this DNA, that is the steps are again repeated. So the region to be amplified, so it will be repeated n number of times, right? That is the T cycles, they can be amplified even up to 1 billion times. So whatever number of copies we need, we can make it by using the polymerase chain reaction. So this is the use of polymerase chain reaction. So then the next step is the ligated DNA is introduced into the recipient host cells. That is the insertion. We saw how the recombinant DNA is made and how the DNA can be amplified, and then how the recombinant DNA is getting inserted into the host cell. So the ligated DNA is introduced into the recipient cells. That is, they take up DNA from the surroundings. So yesterday we saw that is by means of the competent host, right? So the bacteria, actually it is treated with the divalent cations. So there are several other ways of insertion of recombinant DNA into the host cell. So if a recombinant DNA bearing ampicillin resistant gene is transferred into the E. coli cells, the host cells become ampicillin resistant cells. For example, if the DNA, because this is the plasmid DNA, right? And this is the gene for ampicillin resistant. So if the recombinant DNA is bearing this ampicillin resistant gene and it is transferred into the E. coli, so now the host cell, that is E. coli, will become ampicillin resistant because of this gene, right? So if the transformed cells are spread on the agar plates containing ampicillin, so they are having the ampicillin resistant genes now, only the transformants will grow. So those which have this recombinant DNA only can grow in the presence of ampicillin. Untransformed recipient cells will die. So we can differentiate the recombinants and the non-recombinants. Clear? So, but what is our aim finally? That is, we have to get the desirable protein. Whatever from the DNA, that is by means of the gene expression, proteins will be produced. So the ultimate aim is to produce a desirable protein. So if that protein encoding gene is expressed in a heterologous host. So what do you mean by heterologous host or the homologous host? So homologous host means that is from where it originates, the same DNA. But now we are introducing it into a different host. The DNA is introduced into a different host. So that is a heterologous host. And it is called the recombinant protein. Okay. The cells with foreign genes can be grown in a laboratory. So we can grow or we can make these genes in large number of copies. And the cultures are used to extract the desired protein and purify it by using the separation technique. So we can, using the different techniques, we can get the purified genes. And the cells can be multiplied in a continuous culture system. So what do you mean by continuous culture system? That is, the used medium is drained out from one side. So after the synthesis, the used medium will be drained, right? Medium. While the fresh medium is added from the other side. So here the fresh medium is added. So it maintains the cell active and it produces a larger biomass. That is larger biomass since there is more production of the proteins. That is, it yields the more desired proteins. Okay. So are you able to follow the steps? Almost all these steps we saw, right? The first isolation. Then we already studied about the role of restriction endonucleases. So how the cleavage happens. And then after the cleavage, that is the fragments are formed, how they are separated using gel electrophoresis, that also we saw already. And then after the recombinant DNA is made, today the new topic that we saw today is the PCR, right? So here amplification or the production of large number of copies. And after the recombinant DNA is made, it is introduced into the host cell, and even how the DNA is introduced into the host cell. We saw the different techniques by means of the competent host or by means of several other ways like vectorless method, microinjection, and also electrophoration, gene gun methods. So different methods we saw. So after the today, so the main focus is on how the final product is formed. That is how they are made in large quantities by using the bioreactor or a fermenter. Okay, so the ultimate aim is to produce the particular protein in large quantities. So for that, we are using the bioreactor because the cells can be multiplied in a continuous culture system. That is, the medium is drained out from one side and fresh medium is added from the other side. So the cells are maintained in an active state and it results in the production of more number of desired protein. Okay. So what do you mean by a bioreactor? So bioreactor are the vessels, that is, they are the larger fermentation vessels. So in which the raw materials are biologically converted into specific products, or the enzymes using microbial, plant, animal, or the human cells. That is, by using the living organisms, the raw materials are converted into products, whether it is a production of a specific protein or an enzyme with the help of a living organisms in a large vessels. That is called the bioreactor. So they are used to produce large quantities of products. So we can produce it in a larger scale for the industrial production. And they can process about 100 to 1000 liters of culture. So we can easily make the or produce in a industrial scale. And what is the role of bioreactor? That is, it provides the optimal growth conditions. So what are the optimal growth conditions? That is, it provides the proper temperature which is needed for the maintenance of the particular culture, and pH, substrate, salts, vitamins, and oxygen to get a desired product. So to get a product in the proper, or to get a product in high yield, the growth conditions has to be maintained. So the bioreactor provides the optimal growth conditions for the maintenance of the culture so that the yield of the product will be high. So the bioreactors are nothing but the place where the fermentation is happening, right? Fermented, that is with the help of microorganisms. So it provides the optimal growth conditions. So there are several types of bioreactors, but the most commonly used bioreactor are of the type that is stirring type. Stir tank reactor is the common type of bioreactor. So it is usually a cylindrical or a curved base. So this is the structure of a bioreactor. It is a cylindrical or the curved face, right? Cylindrical or the curved base to facilitate the mixing of the reactor contents. So it will be of curved base and helps in the mixing of the reactor, and there occurs in stirrer. What is the role of stirrer? It facilitates the even mixing and oxygen availability, provides the even mixing of the contents in the reactor so that the oxygen availability will be uniform throughout. And there are another type of reactor that is called the sparged reactor. So this is the structure of the sparged reactor, and this is the simple stirring reactor. Okay, so these are the different types of bioreactors. So in both are of stirring type. So it is a usually cylindrical or with a curved base to facilitate the mixing of the reactor contents, and the stirrer, it helps in the even mixing and it makes the uniform oxygen availability. And in some cases, that is, air can be bubbled through the reactor, right? So this type of reactor where the air can be bubbled through the reactor, that is called the sparged stirred tank bioreactor. The other one is a simple stirred. So this is the simple stir tank bioreactor, and this is the sparged stir tank bioreactor. So here the bubbles are actually sparged or sprinkled. So the main difference between these simple and the sparged is here the bubbles dramatically increase the oxygen transfer area. So it is a broader and flat. So it increases the oxygen transfer area in the case of the sparged stirred bioreactor. So increase surface area for oxygen transfer here. Here the stirrer is present, and this stirrer, it helps in the uniform mixing of the content, and also it improves the oxygen availability. Right? So there are different systems that are present. That is an agitator system and an oxygen delivery system for the maintenance of the oxygen, for the entry of oxygen, and a foam control system. And a particular temperature can be maintained by using a temperature control system, and there exists a pH control system, and there are various sampling ports for the periodical withdrawal of the culture, and also the fresh medium can be inserted, right? So this is an example for the simple stir tank bioreactor. So acid base for pH control, and steam for sterilization, and mortar and the foam control system is present, and this is a stirrer which helps in the mixing of contents, and this is the sparged stirred tank bioreactor, where the presence of this bubbles, it increases the oxygen transfer area. So these are the various parts of the bioreactor. Okay, so any doubts till now? Clear? Okay.